Thanks for staying around. So yeah, my name's Sean Su. I'm a research assistant at the Ritchie Lab at Walter Eliza Hall for the Walter Eliza Hall Institute for Medical Research, and we're just across the road from the University of Melbourne. So I'll be talking to you about Glimma, a package I developed for interactive graphics for RNA-seq analysis. And since I'm not assuming everyone here is familiar with RNA-seq analysis, I just want to go into some brief background into what we do and what kind of data we work with. So in a very simplified view of genetics, we have the DNA, which is then transcribed into RNA fragments, which is then translated into proteins. And the proteins are really what make up our whole bodies and what affects our biological functions. So genes, as we refer to them, are basically just small segments on our DNA, and these are reflected by the RNA transcripts produced from the DNA. And RNA sequencing these days is just about sequencing these RNA molecules in order to quantify the amount of genes kind of floating around in our cells, and that's what we're studying. So differential expression is basically just the differences in the expression levels of RNA molecules. From those raw sequences we take from the RNA, we can actually work out which gene they come from, and the result is that we can quantify the expression of certain genes into the table we see on the screen, where we have a bunch of samples coming from individuals which are generally grouped into some kind of treatment or healthy disease groups. And for each gene, we will have a count of that. And that's our expression levels for each of these genes, for each of these samples. And so what we're generally interested in is how particular genes differ in expression, and that's the differential expression part of it, between experimental groups. So we take our big matrix and we fit a bunch of statistical models, and at the end of the day, these two things on the screen are generally what we present to our collaborators. So we do a lot of work, we do our own research, but a lot of the times we're trying to help biologists analyze their data. So on the left is what's called an Mds plot, a mean difference plot, where along the x-axis is the average expression. So each of the points on the screen represents a gene. Along the x-axis, we have the average expression of that gene in our samples. And along the y-axis, we have the log fold change, which is a difference in our two groups of comparison on a log scale. So the highlighted points are all the points that are statistically significantly differentially expressed. And you can see that obviously the bigger their log fold change is, the more likely they are to be considered statistically significantly differentially expressed. So along with that graph is usually this big table of the statistics that we generated, along with descriptions. So each of the rows on this table represents a gene, and with all of that, we ship this off to our collaborators. The plot on the left looks great in a figure, but doesn't really mean much. And the big table on the right is a bit hard to look through. There's tens of thousands of rows that they might have to look through to find genes of interest. So the motivation behind the work I'm about to present is essentially that a lot of times they see this nice, pretty graph and they ask questions about what that particular gene is. It looks like it might be interesting. And we may tell them and send them an email, and a week later they'll come back and ask us about all the other genes on this table, on this graph. So what we really wanted was to be able to make it easy for our collaborators to explore their data and figure out what each of these points are, information about them. We want to be able to link those points with the big table, because looking through the big table is not particularly interesting. And also we want to make it easy for them to actually find the specific genes that they might have been interested in when they started the study. So as a part of this, we had other requirements in mind, and that was to make this be a very easy to distribute visualization. There are certainly a lot of things you can do with Shiny, but as we saw this morning, there are a lot of really cool things you can do, but distribution is still a bit of an issue. And personally, I just want it to be responsive as well. Once you're working with tens of thousands of points, things tend to start getting a little bit clunky. So as a result of this, I developed the Glimma package. It's, it stands for the graphics from Limma, which is a very popular package for RNA-seq analysis that's available on Bioconductor. And Glimma is also available on Bioconductor, and I designed it in a way that if you're already working in a Limma workflow, it should be reasonably easy to change all your plotting calls into the Glimma versions for interactive plots. So really, there's only two plots available in Glimma. The first one is just the Mds plot. This is analogous to PCA for everyone who's familiar, where we take our, so each of the points on this plot represents a sample, and we think of our samples for each of our samples is a data point, and each of the genes is essentially a coordinate. So each of our samples can be thought of as a 30,000 dimensional data point. And MDS, as well as PCA, are just techniques for reducing that dimensionality so that we can represent our data points in a 2D space. And what you're seeing here is on the left, you've got the MDS plot, and on the right, you've got a bar plot of the variance explained by each component of the MDS. And you can very easily toggle between the dimensions you're plotting by clicking on the bar plot. What this is useful for is that usually we only just plot the first two dimensions, and we only go further down if something looks wrong, and we try and discover what each of these components are telling us. So this makes it very easy, so there's no reason not to look further. This makes it very easy to just keep going down the components to try and see if there's other signals present in your data set. And the other feature of this particular plot is that you can put in different groups that you might want to color by, because often there's a lot of confounding factors in your data, and when you look at an MDS plot, depending on how it separates, you can try and infer what the actual dimensions are representing. So here we have the example of group, which is just our experimental groups, and then batch, which is sometimes when they do experiments, they do in different batches, and that can have a significant effect on your data. And this makes it easy to kind of flick through. You can put in as many things as you want and flick through that to see whether any of them become the major source of signal in your particular data set. So the other plot, I've got the MA plot now. So this, it is the MD plot, mean difference plot, MA plot, same thing. The name's been changing around for reasons. So this is just the mean difference plot that we saw before. And what's going on here is that on the left, you've got the mean difference plot. And now when you hover over the points, you can actually get a little tooltip that pops up and tells you about that particular gene. It has all the information. If you click it, it actually brings up the table on the bottom. It takes you to the row for the information for that gene. And on the right-hand side is something that you don't usually see, which is the raw expression levels of that particular gene in each of your samples. And that's also separated into, so you can see that they're in two columns, and that's separating your samples into your two experimental groups. So what this actually allows you to see is sometimes you can see some statistically significantly different genes, and when you actually look at the expression levels, you find out that significance is driven by just one outlier sample. And in that case, you might not want to trust in that particular result too much. So in addition to being able to look around and explore this data set much more easily now, the researcher can also use this little search here and look for the genes that they might be interested in. And that allows them to bring it up. And if you click that particular row on the table, it will actually bring up that gene in your graph. And that's just a really nice way. I think I've had good feedback of people who might go to talks and they see some particularly interesting gene, and then they can bring up their data set and see how that gene behaves in their experiment. And that's actually a really nice feature. So let me tell you a bit about how this was all implemented. because we wanted to make good use of all the existing R packages for RNA-seq analysis, we wanted to keep everything within R. So everything that so when you're using this, all you have to think about is the R part of it. It's compatible with several objects from the Limma and EdgeR packages for RNA-seq analysis. There's also limited DE-seq support. I don't personally use that package, so it may be a bit buggy around the edges, but I have tested basic cases. So once you do that kind of analysis, you can take the results that come out the end, put it into Glimma, and it will show you some nice plots. When I start looking at this, and I think even today, when you think interactivity in R, you generally go and reach for Shiny. That seems to be the go-to package for this. But when I thought about it, when I first tried it out, it wasn't quite what I wanted, mainly because it requires R to facilitate the interactivity, which meant that it locks up your R session. It's hosting a little web server. And also, if you wanted to send that off to a collaborator, you're either having to make a collaborator install R, install Shiny. I think there's RStudio need to be installed as well to get all of that to work. And so that wasn't a very portable solution, in my opinion. And also, the other alternative is to host it on a web server, so it becomes a web app, and that's a lot more stuff to manage. So we really wanted to make it so that you can just send the whole thing off without having to worry about all of that stuff. So yes, R is used to connect up all the data objects from the RNA-seq packages, and it just packages up into a little JSON object. It does a bit of data wrangling, and this was written quite a while ago, so I actually had to write all those interactive plots myself using JavaScript and D3, which it's a very low-level library, and it was mentioned in this morning's talk. Today, you'd probably go for a higher level library, and it'll make life a lot easier. And it's actually quite nice, even when you're writing things in JavaScript, writing things in HTML, you can actually hide all of that in the inst folder of an R package, and when you need it, you just copy the whole thing out, and no one ever had to know that there's JavaScript going on anywhere. So the output of all of this is just 2 folders and a file, or multiple files, depending on how many of these you decide to draw. And you can zip all of that up and set off to your happy collaborator to investigate their data set. And all it requires is that they have a modern web browser, which means not Internet Explorer 7. Oh, I'm ahead of time. So yeah, in summary, I think that Glimma produces interactive plots that are very nice for helping you explore and discover things in your RNA-seq analysis. In general, I don't think anyone needs convincing that interactivity is a great way to increase the information density in your visualizations. And I think that it's a very nice thing to try and link interactions between plots to bring up information that people might otherwise not look at and neglect. And as part of future work, what I really like to do is make use of the more modern tools. So taking things like Plotly, and if anyone went to Carson Sievert's talk about interactive web graphics, there's a lot of development in that space now that makes it a lot easier to implement what I have without having to go down to a very low-level code. I'd like to create some HTML widgets, which you can actually embed into the R Markdown reports, which would be very nice. I've had requests to make my current plots embeddable into HTML widgets, which I don't really want to do because that tends to get squeezed down into a little column, and I feel like my plots deserve more space than that. And I would like to add some visualizations and support for single-cell data objects, because it is already with RNA-seq, we're pushing the boundaries of performance and lagginess with a limited number of points. Those are growing rapidly now that single cell is becoming more popular, so there is a lot of performance tuning that needs to be done to make that feel fluid and usable. And also the data is actually quite a challenge in terms of getting everything compact, because those HTML pages and the JavaScript, they may go up to around 200 megabytes these days, and I'm having trouble sending those to people on email now. So, oh, thank you for that. Any questions? There is already a XY plot in there, and you can make your own. Yeah, but I could. It's not too difficult to actually just wrap it up. Yeah.
People who really appreciate the other point click, I just want the high level one.
Yeah, I mean, so the MA plot is sort of kind of an alternative to the volcano plot that we highlight to push onto people. So,
Yeah.
but
I know you guys like it, but I think a lot of people like volcano plots.
sure, that's yeah. It's been on a to-do list for a while, to be honest.
How much D3 input have you input into those interactive graphs that's been produced by Glimma?
How much D3? I mean, it's not really using any, like, D3, I don't know if you've used it much. It draws circles and lines. So it's hard to say how much D3, you know, it's a few hundred lines of code, but that's just how much it takes to draw a scatter plot. So it does help you handle, the actions, so the events hover and clicks very nicely. It's not.
I can say it's like those interactive plots basically use Glimma to produce the graphs individually, and D3 helps to interact.
Oh no, so all Glimma is really doing is shipping out the data from R. So it's taking the R data objects and making it into a JSON format and then writing it out into a little JSON file. And then there's kind of a whole separate part of it it's called Glimma.js. It's not documented, poorly commented, but it's holding together. And there's a lot of JavaScript code. So D3 doesn't handle much for you. Like D3 is a library, and I've used it. It's essentially like grids in R. And I had to write a lot of D3 code to actually draw all the plots that you were seeing. Nice.
If there's no other questions,